my pleasure to introduce a friend, Daniel Colon-Ramos, from our medical school. He was born and raised in Puerto Rico. Somehow ended up at Harvard after that. He's going to tell you how that happened, right? He got his bachelor's, went on and got a PhD. Was that at Duke or? It was a Duke, yeah, Duke, and then he did a postdoc at Stanford, and he's been here since I think 2008. Yep, in our medical school. So he's got had a fascinating career. He's going to tell you all about that, and he's going to start Science Saturdays off this spring with a bang. Oh, it didn't work, Marcus. Damn.
Right. Hopefully the talk will be better.
Hopefully that'll work better. Yeah, yeah. So join me in welcoming Daniel.
Excellent. All right, thank you. And I want to begin by thanking Kurt for the invitation, and also giving a hand of applause to all of the volunteers and the budding scientists that are here. So on a cold Saturday morning learning about science. So let me tell you a little bit about what I'd like to do today. I'd like to tell you about how we use C. elegans, which is a tiny worm that we have taken a picture of here. You can see it here. That's an embryo of C. elegans. We put it like a question mark. How do we use it to understand how the brain works, how memories work? And before I start, I'd like to mention that for the parents, if you're interested in these topics, or if you want to show your kids more videos about science or how do we do science, I have a Twitter account where I usually tweet about these topics. And also there are a number of videos online where I've given a few interviews in iBiology and with Dan Rather on these topics of basic research. But today, what I'd like to do is I'd like to tell you about, first, how I became a scientist. Like, what was it that brought me to science? And what scientists do. So as a kid growing up in Puerto Rico, I liked science, but I didn't know any scientist. So I wasn't really sure what scientists did. And I like to tell you about what scientists do and how they do it, and then how we use a tiny worm to study something like a brain. So how and why. And I'll end by giving some advice to budding scientists or aspiring scientists in the audience, which I see a lot of. And there's going to be one theme in my talk that I'm going to be coming back to, which is very important to how scientists do our work, which is that I like to explain to you how we can use small animals to ask big questions. I'll be coming back to that. Like, how is it that we can use a worm to understand something like a brain? So, like Kurt mentioned, I was born and raised in Puerto Rico, and when I go back home and when I leave kind of the circles of scientists, I'm frequently asked, how did you become a scientist? And I like to answer that question by saying that we are all born scientists. And I'm a scientist, so I did an experiment to test that. I did an experiment to see if we're actually all born scientists, and here are my results. So these are my experimental subjects. These are my daughters when they were babies. I have triplets. This is Keisani, Alexandra, and Beatriz on their very first trip to the beach in Puerto Rico. They're actually doing an experiment themselves. They're learning. This is the very first time they have been on a beach, so they're the very first time they have seen sand. And you can see Keisani is kind of like going and grabbing the sand, and Alexandra is breaking the sand clumps in her fingers, and well, Beatriz is eating the sand. So they're actually using their senses to understand how, what this new thing called sand is. And I'm going to use this as an example of how scientists learn, because what they're doing is not unlike what I do every day. Not that much, but a lot of exploration using my senses. So the first concept I'd like to highlight is that how we learn is context dependent. And what I mean by that is they're learning about sand because they're sitting on a beach. If they were not sitting on a beach, they wouldn't be learning about sand, right? So for example, here's another picture when they were babies. And here, this is in New Haven in the fall, next to a bunch of leaves, and you can see now they're eating leaves, right? So they're learning about what they have around them. This is important to how scientists learn. We learn depending on our context. Also, we learn from each other. So in this situation, Keisani was in the middle, and Beatriz and Alexandra were in the corner, so she couldn't actually grab the leaves like her sisters, but she saw how her sisters grabbed the leaves, and we're putting them in their mouth and then spitting them out. And in that way, she learned that leaves were not edible. Okay? And let me just make an aside here and say, look, these are experiments done by experts. Okay? So kids out there, don't go and have triplets to do these experiments. Okay? This is only for scientists. So we learn from each other. We don't have to go through the experience ourselves. And what we learn is widely applicable. So many years later, and this is a proud achievement as a parent, they learned that none of these things are edible. Here they are years later in a pile of leaves, and you can see they're not eating leaves anymore. So this is actually how we produce knowledge, how both humans and human scientists, how we actually generate knowledge. So scientists don't do something that different from what kids do. What we do is that we produce knowledge by learning from each other, by learning dependent on context, and then by applying what we learn. So what I mean by applying what we learn is note that they don't need to taste the sand in every single beach that they go to know that sand is not edible. Once they know sand is not edible, they'll apply that knowledge to every context. And that's what scientists do too. We generate one set of concepts, and then we apply them widely. And that's important to how we generate knowledge. And I like to underscore here that because of these concepts, where scientists are from matters. Because the context that we're from matters in inspiring the questions that we're interested on. While the knowledge that we generate is universal, what we learn and the context influences what knowledge we're going to generate. And I'd like to share with you how my context, being a Puerto Rican, influenced my interest in neuroscience. And I'd like to share that by sharing another story here, which actually starts with this beautiful video of baby sea turtles being hatched in a beach in Puerto Rico, not far away from where I was born and raised. And I'd like you to notice two things. These turtles are just hatching, and they're all going in the same direction. Actually, they're going towards the ocean. They're beelining it towards the ocean. So how does a turtle know that it needs to go to the ocean after it comes out from the egg? It hasn't been to turtle school. Who told the turtle it needs to go to the ocean, right? So those are questions that I had as a kid. And there's something really amazing that's happening here that's invisible to us, to our eyes, but very important, which is that these animals will remember this beach and they'll come back to it to lay eggs on it. So here I like to underscore two things, right? These turtles have a behavior from the moment that they're born that they know that they need to go to the ocean. That's what we call a hardwired or innate behavior. It's an instinctual behavior. But they also have a memory that they're forming about this place, and they'll come back to it. And these behaviors are actually underpinned, they're formed by the brain of these turtles. And a question that you might have is, how do they know what to do? And let me break that down, because this is actually really incredible if you think about it. So here's the map of the world. This is where Puerto Rico is. And some of these leatherback turtles, they're born like babies over there, and then they're going to grow up to be the size of a car. They grow up to be about 9 feet long, like a Volkswagen. And they'll travel the world's oceans. From that little island of where I was also born, they'll go everywhere in the world. And then when they have to make the most important decision in their lives, which is where they're going to lay their eggs, somehow, in a way that we don't understand, they'll find their way back, not only to the same island, to the same beach that they were born. So that is actually pretty amazing, right? So if you ask yourself, how is it that this animal can do that? All I can say about that is nobody knows the answer. Nobody knows how this happens. And I want to underscore that for a number of reasons. When I was the age of many of the kids that are here in the audience today, you go to school and somebody asks you a question, and then they ask you a question and they give you an answer. And then they ask you a question and then they give you another answer. And when you're a kid, you get the impression, well, all these questions have answers, and the only person that doesn't know the answer is me. But the truth is that for most of the questions, the most interesting questions in nature, we don't have answers, period. And what scientists do is we figure out what the answers are. That's what our job is. And there are a lot of questions out there that are unanswered. And I'm going to be emphasizing some of them to see, because someone here, someone in this audience, will actually go on and answer some of those questions. So we do know some things, right? We do know, for example, that the brain is important for forming memory. So the turtle's brain is important for it to know where it needs to come back to. And we know that one human brain, this is a human brain, has about 100 billion cells or neurons. So how many people here have heard about the word neuron before? Show of hands. Okay, so neurons are the cells that make up a brain. And in each one of your brain, there are 100 billion neurons. So how much is 100 billion? Just to give you an idea of how many cells we're talking about, let me make an analogy. There are more cells in your brain than there are stars in the Milky Way. Okay? So when you stand, like when you go camping with your parents, and you're looking at the immensity of the stars of the Milky Way and thinking about the possibilities that are out there, think that your brain is a reflection of the possibilities of the galaxies. You know, your brain there are as many infinite possibilities in your brain as there are stars in the Milky Way. So look at it as a mirror of your own possibilities. And the neurons that form the brain, they connect to each other. Here's a cartoon diagram, like a drawing of how these neurons will look like. You have the cell body, and then you have extensions of these neurons, and they're talking to each other all the time. And they talk to each other through electrical signals. That's happening in your brain right now as I speak. So when I'm speaking, my vocal cords are actually creating sound waves that are moving tiny hair cells in your ears, and somehow, in a way that no one understands, that is creating thoughts and memories and emotions in your own brain. And what neuroscientists do is we try to understand how that process happens, how these neurons communicate with each other. And here are some of the most interesting but unanswered questions in neuroscience. How is it that the brain gets built during development? So you have 100 billion neurons that have to be connected to each other precisely so that the brain can function. How does that happen? How does the neuron know who to connect to? Another question that we don't know the answer to is, what is a memory? So anyone here knows what a memory is, in the sense that you can think right now of the face of your mother or a loved one or your best friend, and an image comes to your head, right? But no one can tell you how. What is that image? What does it represent? How is it encoded in the cells in the brain, in the molecules that you have in your brain? And then, importantly, what happens when the brain breaks down? How many people here in the audience know of or have heard of people that suffer of diseases like Alzheimer's and Parkinson's and autism, right? So all of those are diseases that are rooted in the functioning of the brain. And some diseases like Alzheimer's and Parkinson's happen when some of these circuits that form our memories start breaking down. Now, it's going to be hard for science and for medicine to figure out how diseases like Alzheimer's work and how it results in losing memories if we don't know what a memory is, right? If we don't know what we're looking for, how are we going to find it? So as scientists, what we do is we ask, well, what are these things? And again, I want to underscore all of these questions, they don't have answers. So when you go to school, maybe they'll ask you questions that have answers. Maybe you'll do experiments that they'll know a result and you're supposed to get it right. This was, I'm telling you, they don't have answers. And the reason I'm emphasizing that is because maybe some of you will find the answer to it. So that's one of the things that I find very stimulating as a scientist. And when I was a kid, thinking, wow, I can use my brain to contribute something that no one has known before. That's one of the things that I find really exciting in science. So, okay, so I've been talking about the brain and turtles and all these cool things. And this is the part of the talk, the awkward part of the talk, that I have to explain to you how we do it. And I say the awkward part of the talk because when I say what we do, people are like, wait, what? Like, you do what? And so we do it by using a tiny worm. Here's the picture of the worm that we use, which is called C. elegans. It's a nematode. It's not that big, okay? It's about the size of a comma in a sentence. It's tiny. It's transparent. It doesn't cause any disease. You can touch it with your fingers. It lives anywhere. It doesn't have any economic value. So why are we using a worm to understand how a brain works? And people actually, some people get annoyed by that. They're like, are you telling me that you can understand my brain by using a worm brain? How does that work exactly? You just spend a bunch of time telling us how wonderful the brain is. And so here are some of the frequent questions, like, what? How you can use a worm to understand a human brain? And why play with worms and not work on something more important like disease? Like, you're using a bunch of money to pay people to understand these worms. Why don't you go and work on something more meaningful? And okay, so I get that knowledge is cool, but is it really useful? Like, what are you gonna do? You're gonna build a worm robot? Like, what are you gonna do with this knowledge, right? So to explain that, I'm gonna do a detour here. Let's leave the brain aside and the worm aside for 1 second, and I wanna explain to you how science works. And I'm gonna do it by using some historical examples that I think are pretty cool, and I'm gonna ask some questions to the audience too to engage you. So I'm gonna use some discoveries to explain how science works, and I'm gonna end up explaining how and why we use worms. But to do that, I'm gonna start with 2 questions. Let's suppose that you have $1 million, and somebody tells you you can use that $1 million to understand and cure a disease like Alzheimer's or cancer, or you can use that $1 million to understand a question like how you build a worm, essentially how you go from a single cell to a full worm. Honest question, show of hands. How many people will use the $1 million to do some experiments where you think it will result in a cure to cancer? Okay, most of you, and that's reasonable. How many people will use the $1 million to understand how a worm gets built? A few of you, okay, good. So in the next 10 minutes, I'm gonna ask you another question like this, and in the next 10 minutes, if you pay attention, you're gonna understand why people can do these experiments and contribute to understanding how diseases like cancers and neurodegeneration work. Here's my my kind of like PG-13 part of the talk. Kids, you might wanna cover your ears. How bacteria exchange genetic information. How do they have sex, so to speak? So how many people will wanna know the answer to that question versus, say, build the most important biopharmaceutical company that you can build? Okay, show of hands, who will use the $1 million to build the most powerful pharmaceutical company you can build? To develop new drugs, okay? Who will use the $1 million to understand this question? Okay, see, they're very interesting, very interesting. So, okay, so again, I'm gonna explain how these discoveries led to one of the most profound biopharmaceutical discoveries that we have had. So let's start with this question. How do you build a worm? Okay, so this is a pretty fundamental question. So in development, all of us, every single person in this audience, started as a unicellular organism at the moment of conception. So you have a sperm that goes inside an egg, and then you have a single cell. And from that single cell, you develop a whole organism. How? People don't know. But a group of scientists about 25 years ago decided they wanted to examine that question that is very interesting by using this worm. And what they did is that they sat down and they counted cells. They just looked at these cells. And they said, okay, from a single cell, you get 2. And from 2, you get 4. And from 4, you get 8, and so on and so forth. And they kept track of every single cell division as the worm forms. And they noticed some patterns as they looked at different animals. They noticed that certain cells, which I'm going to represent here with these balls, certain cells always became nerve cells. The same cell in different organisms always became a nerve cell. Then other cells always became muscles, and other cells always became gut. And this led to a very important concept in development called cell fate. These cells are destined or fated to become nerve cells. These ones are destined or fated to become muscle, and these ones destined or fated to become gut. And this open up all sorts of questions about what are the signals, what are the molecules, what are the instructions that tell the cell what it's going to become so that it can form an organism. But then they observed something that didn't make sense, something that was weird. What they observed was that there were certain cells that were always destined to die. And the reason this didn't make sense is because people were thinking that the way you get life is by growing and multiplying, not by killing and dying. So why is it that certain cells are always destined to die? Like as part of a developmental program, not because they're sick, not because they're damaged. They were destined to die the same way that this cell is destined to become a nerve cell. So they started asking those questions, and then they realized that the way that you grow an organism is not only by dividing and multiplying, which is very important, but also that death is a very important part of life. And let me give you an example to explain that. If you look at a duck, at the digits of a duck, they're webbed, right? They have tissue in between them. And if you look at the digits of a chicken, they don't. The reason that the digits of a chicken don't have webs between them is because, well, they need them to be able to carve in the ground rather than swimming. And the webs in between those digits, they actually die so that you can sculpt out the digits of the foot of the chicken. Does that make sense? Actually, our own digits were not formed by cells growing out like columns. Instead, our hands were kind of like that, and they were webbed when we were embryos, when we were fetuses. But then, all these cells in between died, and that's how we can have our digits. So a way of thinking about it is the same way that in order to get shape out of a block of marble, the sculpture has to like carve away pieces of the marble to develop some shape. When our own organs and our own bodies are forming, cells need to die. So this is a pretty big discovery in developmental biology. It's a pretty big one. And then the scientists then went on to identify the molecules that are the signals that tell to the cells how to die. They realized they were really important molecules that were essential for normal development. When those molecules break, if, for example, you have a cell that's supposed to die and it doesn't die, that cell starts multiplying abnormally. And when it starts multiplying abnormally, you end up with tumors. And when you end up with tumors, you can end up with problems like cancer. So the mechanisms that they figure out in the worm are actually very important for understanding cancer. Then, if you have cells that are not destined to die, but these molecules are always active, they're killing the cells that are not supposed to die, you end up with problems like neurodegeneration, because you're going to lose tissues that you were not supposed to lose, like nerve cells. So for these discoveries, that group won the Nobel Prize. Okay, so this is actually a pretty important and fundamental discovery. Now, you open the newspaper, you will read that there's a group of people that won the Nobel Prize for discoveries that they did on cancer and neurodegeneration. But I want to remind you that they were not looking at questions of cancer and neurodegeneration. They were looking at a fundamental question in biology. And the fundamental question in biology they were asking was, how do you form a worm? Does that make sense? This is very important because this motivation of how is it that you start with the question in biology, you cannot answer these big disease questions if you don't understand how the fundamental processes work, right? So that's one of the aspects that I like to underscore here. Now, let's go to this example here of how bacteria have sex. So in this example of how bacteria have sex, this guy actually is a real-life person. He's a scientist. His name is Herb Boyer. He worked at Yale as a postdoc. And Herb Boyer became interested in how bacteria exchange genetic information. And he discovered that one of the ways in which bacteria exchange genetic information is that they have proteins that are able to cut DNA. Like you can cut DNA, and there are other proteins that can paste it. And then he had an idea. He thought, wait a second. If you can cut DNA from one organism and paste it into another one, can you trick a cell from one organism to express a protein from another organism? So how many people here are or know of other people that are diabetic? A lot of you, okay? So until the 90s, if you were a diabetic patient, the way your insulin will come from pigs. Okay, insulin was harvested from pigs. And pig insulin, it came from pigs because pig insulin is very similar to human insulin, but it is not human insulin. I don't know if, like the parents probably remember this, but there were all these conversations about how if you get like insulin from pigs, are you bringing retroviruses or other viruses also from the pig into the patient, right? So Herb Boyer had an idea, which was, can we take the gene that codes for insulin and use the genetic tricks of cutting and pasting DNA and trick a bacteria cell to make insulin? And he did that, and that resulted in the birth of the biotechnology industry. So the specific company that he started is called Genentech, but this is a $430 billion industry right now. Now, if you open a newspaper, you will read that the scientists developed this $430 billion industry by developing insulin to treat diabetes. And you will think that that's how he started thinking about it. But I want to remind you that the question that he started with wasn't, how do you treat diabetes? It was, how is it that bacteria have sex? Now, these two examples are arguably extraordinary because they have extraordinary results. But they're not extraordinary in the process because this is how science actually works. This is how scientists actually develop their own ideas. They start with curiosity-based research, and then they build that up to these discoveries, right? So for all of you that live in New Haven, Herb Boyer donated a building that's called now the Boyer Center, which many of you have probably seen in Congress Avenue. So I'll finish with one last story, and then I'll go back to how we use the worms to study how the nervous system works. So how many people here remember Superstorm Sandy? Maybe many of the kids were young, but you remember it? Do you guys remember that there was an old oak that fell in the New Haven Green? Did anybody remember that? Or was that lost in losing power and all that? So there's this oak that was planted in the New Haven Green like 100 and some years ago, back like I think in the 1900s. When this oak comes down, in its roots, what do they find? Do you guys see this here? That's a human skeleton. You can see the teeth here. The eyes will be around here. This is the back part of the skull. Here's the rib cage. You see it? Okay, how many people here knew that the New Haven Green was actually a cemetery? Okay, so the New Haven Green is a cemetery. So when you're actually standing in the New Haven Green, you're actually standing in what used to be a cemetery. There are a lot of corpses right underneath you there. And this tree had its roots around what used to be a common grave. And it was a common grave that was built in the 1800s because there was an epidemic of yellow fever in New Haven. And there were so many people dying of yellow fever. How many people here have had yellow fever? Nobody. Okay, there's a reason for that, which I'll explain. But back then, people were dropping like flies. There were so many people were dying, they had to build a common grave for the people that were dying. And this is one of the unfortunate people that lost their life in that epidemic of yellow fever. So I wrote an article about this, and this happened the day of Halloween. I don't know if people remember that, but it was the day of Halloween that this tree falls down and a skeleton comes up. So this past Halloween, a century-old human skeleton resurfaced in the New Haven Green in Connecticut after Superstorm Sandy. It reminded everyone that what is today the city's Central Park was once the town cemetery. It also reminded some of us that in an earlier time in New England, life expectancy was about 40 years. Do people know what life expectancy? How many people know how life expectancy is the average number of years that you're expected to live when you're born? It doesn't mean that everyone died around when they were like 40 years old. It just means that a lot of people die young because we didn't have the medical advances that we have right now. So life expectancy in New England, when this person died, was about 40 years. Now it's 78 years. Okay, so what happened between then and now? And let me start by saying that between the Stone Age and the 1900s, for 40,000 years of human history, life expectancy did not change. Isn't that amazing? 40,000. So there were empires that came and went. People built the Eiffel Tower, the Great Wall of China. Life expectancy worldwide did not change for 40,000 years. And it was about, in the world, it was about 30 years of age. In the US, it was 40, but in the world, it was 30. So, and right now, the world life expectancy is 72. It doubled. So what happened? What happened at that time? So if you look back at medicine, at Western medicine at that time, the main thought, the way that people thought about medicine, was that we had these different humors that controlled who we were. And I'm not going to go into details about how that worked, but let me just tell you that that's not how the human body works, okay? And the reason, this is a good example that I like to use. This is George Washington in his deathbed. So how many people here know what George Washington died of? Few people. What do you guys think George Washington died of? Yeah.
He died of a throat infection.
Yes, very good. He died of a throat infection. He was 68, I think, 60, something like that. And he died of a throat infection. But actually, the cause of death, they didn't have autopsies back then, but people think that the cause of death wasn't the infection itself. He was bled 5 times. They took over 5 liters of blood out of his body to cure the infection. Why would you take 5 liters of blood out of a human being to cure an infection? Well, how do we treat infections today?
Well, I don't know how they treat infections today, but I think that taking out blood was to get rid of the bad blood
Exactly. So back then, they thought that there was bad blood. And for you to cure an infection, you needed to get rid of the bad blood. That's exactly right. That's what they thought back then. That's not how it works. If you have an infection today, how do you cure it? What do you do? Antibiotics. That's right. So why didn't they use antibiotics back then?
They didn't have any.
They didn't know. They didn't even know that bacteria cause disease, right? So actually, doctors back then, have people seen this image? Have some of you seen this image? What is this? What's that?
The plague.
That's an image of the plague, but this character here, who will wear that mask? A doctor. This is a doctor. It's now a symbol of death and the plague, but this was a doctor. And you know why they wore that mask? Yeah.
There was herbs inside that mask, so they didn't get the plague.
Excellent. So they will wear this mask so that they could put a bunch of herbs there to block away the bad air. Malaria, the disease, is called malaria because in Italian it means bad air. And the reason is because they had no idea that it was caused by a parasite or that it was transmitted by mosquitoes. All they thought was, there's bad air, so I'm going to block Now, that doesn't work because that's not the root cause of the disease, right? So how is it that humanity ended up advancing life expectancy, discovering microbes and things like that? And it was through basic research. So what changed? What changed was that there were curious people, like this guy here, this is Leeuwenhoek, who developed the first microscopes, and then they discovered, wait a second, what I see is not all there is. What I see is just a fraction of what's going on, and there's a whole universe of microscopic organisms out there that I cannot see with my naked eyes. I need microscopes to be able to see them. And then after he made that discovery of microscopes and microbes, there were people like Pasteur that made discoveries of sterilization, and later Alexander Fleming, who made the discovery of antibiotics, like it was mentioned here, right? So, but let me underscore this. If we don't know about microbes, you know how we cure disease? We bleed you to death. So with that, I like to underscore that even Western medicine that we're so proud of, without science, is little more than witchcraft. Okay, actually can do more harm than good if we don't know actually what is going on. So this is the end of my detour. I'm going to come back to the brain and answer this question. Why do we use an animal like this to study the brain? Well, part of it is curiosity-driven research. We're just interested in how it works. The same way that the people that made the discoveries that were very important for cancer and neurodegeneration made the discoveries by looking about how a worm forms. So we're interested in how does this animal think. The other part is that nature, when it comes up with a solution to a problem, it uses it over and over again. So the nervous system that makes this animal think and behave, if you look at the building blocks of that nervous system, it's not unlike the building blocks of our own nervous system. Yet we don't understand how it works. So we think that if we can understand it in this organism, we can understand it in our own nervous system, in our own minds, and how it breaks down in disease. And the last part, which I raised earlier when I was showing the picture of my kids, is that what we learn, we can apply it widely. So what we learn in this organism, the fundamental aspects, we can then apply to other things. But one of the really cool things that we can do in this animal is that we can actually, because it's transparent, we can label it like a Christmas tree. We can look at all the different cells that form part of the animal. Here's the head and the brain and the different neurons that form it. And then we can make images like this. This is actually a picture taken by my friend Mark Hammerlund, who's also a professor here. And look how beautiful he can label this animal. He can actually label neurons that make a neurotransmitter called acetylcholine in red, and another set of neurons that labeled one that makes GABA in green. And you know, it actually looks gorgeous to me, right? But the important thing is we can see single cells. So how do we do this? How can we do this? And the way that we can do this, and I want to explain again how we make discoveries, is because a few years back, scientists, a different group of scientists that don't study the brain at all, had also a curiosity-driven question, which was, how is it that this jellyfish, which is called Aquaora victoria, how is it that it glows? That was the question. They didn't want to cure a disease. They didn't have like big aspirations. Just how is it that this animal emits beautiful fluorescent light? And by studying how it did it, they ended up discovering, this Japanese scientist, Osamu Shimomura, ended up discovering a protein that is important for this animal to fluoresce, which is called green fluorescent protein because it fluoresces in green. And then this other scientist at Columbia University, Martin Shalfie, put it inside worms, and he could actually see with this fluorescent protein the structure of the whole nervous system. And then there was a third scientist, Roger Chen, who then took it and modified it to make it all sorts of different colors: red, yellow, blue, green, etc. And actually now we have all these different variants: GFP, which is green fluorescent protein; YFP, which is yellow fluorescent protein; and M-Sherry, which guess what color it is, right? It's red, right? So, and this is actually a picture that was done with bacteria. So this is a bacteria plate, and you have different bacteria that are expressing these different proteins, and they have actually plated them in different ways to make this image. But these are all bacterial colonies. Isn't that amazing? So, and then, but importantly, you can label single cells in the nematode, in the animal C. elegans. So this is how we can actually generate this type of image. We can use fluorescent proteins, we put them inside the animal, and then we can see these cells. And what is the importance of seeing these cells? Well, because we can see single cells with these fluorescent proteins with microscopes that my lab have developed with other collaborators. And here I'm putting pictures of collaborators because when I was a kid, I had the impression that science was this very lonely process of like thinking and coming up with ideas, and nothing farther from the truth. It's actually a ton of fun. You get to work with some of your best friends, and it's actually fantastic. And I get to meet friends and get together and think about how to answer questions. And one of the ideas that we had was to build this microscope, which allows us to take movies like the one I'm going to show you here, where we put a fluorescent protein inside an embryo, and we can see the brain forming. So these are the very first cells that form a brain in a C. elegans embryo. You can see them growing out and forming a ring there. And now the animal starts moving, and all these other cells join. And now this is the beginning of the brain of the animal. And I'm showing you this accelerated, but we can actually go frame by frame of the movie and kind of make up this story of how is it that these cells are coordinating, are talking to each other to be able to build a brain, which is one of the important questions that I told you at the beginning that lacks an answer, right? If you're interested in this and you want to see other cool movies, there's some iBio seminars in which I have explained this in more detail. But for the purposes of now, I just want to mention we can see the development of the nervous system as the animals are growing. And the other thing I want to come back to the turtle that we can do is that the turtles, I told you that they grow pretty big, about the size of a Volkswagen. And then they will come back to the beach in which they were born, and they'll remember that beach. And we don't know how that happens. And nematodes, well, they don't travel the world and go to different beaches, but they also form memories. They also remember things. Let me show you one of them. So the nematodes, unlike us, they don't have an innate preferred temperature. So humans, we always like the same temperature. Even people that say, oh, I like the cold, they don't like the cold. I mean, they like to be in the cold with a lot of jackets on top of them. But we need to have the same temperature, about 37 degrees Celsius, us, humans. The worms, not so much. Between 15 degrees Celsius and 25 degrees Celsius, they don't care that much what temperature they're at. But they remember if they were in a pleasant condition at that temperature. For example, what do worms like? These worms, they like to eat bacteria. So if you take a worm and you feed it at 15 degrees Celsius, and then you put them in a temperature gradient, that animal will remember that temperature and come back to that temperature when you put them in a gradient. Look at this. So this is a gradient here. These are a lot of little worms that we're putting. We train them to like the cold temperature, and what are they all doing? They're moving towards the cold temperature, okay? Now we can take these same animals and retrain them. It takes about 4 hours for them to learn the new temperature. And if we put them now at 25 degrees, look at what they do. Now they all move to the warm temperature. Here I colored them blue and red to represent the experience that they had. But these animals are identical to each other. These are the same animals. The only difference between these animals and these animals is the previous experience. So you can see that they can learn a temperature that they like. And then, so we can represent it like this. The ones that were grown in the cold, they go to the cold. The ones that were grown in the warmth, they go to the warmth. And then we can ask, well, what changed in their brain? What changed here and here that these animals now remember different temperatures? What is a temperature? Like, what is a temperature in the brain of an animal, right? So that's the question that we had. And then we have developed ways of, this is a movie, let me see if it plays. Okay, so here you can see a worm. and it's moving and we're tracking it, this is a zoom in, you were seeing a single cell in the brain of the animal, and we're tracking how that cell is changing activity, essentially how the electricity is changing through that cell as this animal is performing the behavior of thermotaxis. And there's a lot of complex information here, so let me boil it down in this experiment I want to show you. So just to show you the kind of experiments that we can do. We can look at single cells in the brain of live animals by using these fluorescent markers from jellyfish, right, that I told you about. We can build microscopes when we can change the temperature very precisely. And then here, I'm going to use this as a placeholder to explain the experiment, and then I'm going to show you the result. So what we're doing is that we're giving it a temperature ramp. We're going warmer and warmer and warmer, kind of in oscillations. And this line here represents the temperature at which the animal was in the experiment. The result is going to be here, and what you're going to see is us tracking a single neuron. This is the cell body, this is the axon, and the dendrite that it uses to feel the temperature. And let's see what the animal does. That's the cell body. The animal is alive, so it's moving. Nothing is happening. Then it hits the temperature that the animal was seeing, and it's almost like a sing-along. You guys see that? Like every time the temperature increases, this increases in fluorescence. Let's look at it again. Nothing is happening until it hits this point here, which is the temperature that the animal was at. There. Boom, boom. You can see the nose moving here. Boom, because the animal is alive. So what this tells us is a few things, but I'm going to ask the kids in the audience to see if they can help me interpret this result. So first question, what does it tell us about this red line here? So what happens over here? Is the animal responding or not responding? Not responding. Okay, so it tells us that the responses really start at the temperature that the animal was before the experiment. What is another experiment you like to do? Is that a fixed thing? Is that always the same for the temperature, or can you change that? Right? That's a question I'd like to know. I'd like to repeat this experiment and change the temperature that the animal was at and see if I can change this threshold, move it up or down. Does that make sense to people? Would you like to do that experiment? Right? Okay, so we did that experiment, and it turns out that this temperature that the animals start responding to, it changes dependent on the experience that the animal had before. Okay? So then what does it tell you about the responses over here? So what is the animal responding to? What is this cell responding to here? It's responding to warming above this temperature. So what this neuron is essentially doing is telling the animal, you are warmer right now. You're moving to a warmer temperature than you were before. But that temperature, this one, it changes as the animal is navigating the gradient. So now, doing this type of experiments with a jellyfish protein in a nematode, we can start understanding how memory works. Because we can start piecing together the puzzle of how this animal can conceptualize an abstract concept like temperature. What is temperature for a neuron? Right? The neuron is not a thermometer. How does it know how to interpret temperature? And to pass it on to the other circuits of the brain, and to tell the animal which temperature it should go to. So we can start doing those type of experiments with this. Does that make sense? Any questions about that? Yes?
So if it's not responding as you're approaching the temperature until you hit the temperature, once it crosses the threshold, I might expect it to respond negatively so that it could go back to the temperature. But
Right.
It doesn't look like it does that.
So that's a great question. So what happens when you don't have information here, right? So how does the animal know when it's going colder? Like I told you how the animal knows when it's going warmer. So this gets to an important point of science, which is that with this sensor, we can see how the animal is responding to the warmth, but we cannot see when the animal is not responding, right? There's some things we cannot see with these sensors. We cannot see other cells. We cannot see these other responses. But what we can do is that we can measure electricity. We have ways of measuring electricity in that neuron. And we know that when the animals are going colder, actually this neuron, what it does is that it instead of communicating more electricity, it reduces its capacity to communicate electricity. So there's information there that we're not actually capturing in this experiment. Does that make sense? So the important concept for all the budding scientists out there is that some experiments will give you some information, but no experiment gives you all of the information. So you always have to supplement with different experiments to be able to learn that. Any other questions? Okay. So other things that we can do in my lab is, for example, we can watch what I call the dance of the molecular machines that produce energy. So I'm going to show you here. This is a cell that has here in green, we have molecular machines labeled that are known to produce energy. And this is a video where we can make the animal energetically stressed, and we're going to see that they're going to start forming these here. They start forming these clusters, and then we relax the energy. So now they have energy, they go away. We energetically stress them, they form them again. We reduce it, and they go away. So let me play that again and explain it. I explain what we're looking at. So here we go. Okay, so what we're doing here is that we are telling the animal, we're giving the neuron a stimuli in which we energetically stress it. And then we see this machine that makes energy. When we energetically stress the neuron, we see this machine that makes energy form and respond by making more energy. So we are controlling a dance of molecules inside this cell that come together, they cooperate to make this energy machine. So right now the neuron is in normal conditions. It has no reason to make these machines. Sometimes some of them are formed here, but in many places they're not actually formed. And then when we put them under energy stress, so now we're telling it, you have to make this machine if you want to have energy. This machine self-assembles, and boom, now you can see it. These dots represent the machine forming. Even in these areas where the machine was kind of there, you can see that it becomes tighter. And it becomes tighter because the machine is forming. When we remove the energy stress, now the neuron doesn't have to form these machines. The machine dissolves. It doesn't form anymore. Then we induce the energy stress, they form again. So doing this type of experiments, we can see how the molecular machines inside these cells are actually responding to even the animal's thinking, needing energy for thought processes, and things like that. Like, how are they responding to be able to create thoughts and experiences, sensory experiences in the animal? And I'm going to finish by saying what we have learned, and then I'm going to move to the last part, which is just some advice for the body scientist. So what have we learned so far? We learned how neurons find each other and how they connect using the microscopes that I show you. We have learned how neurons maintain the connections during growth. We have learned how they modify their connections during learning and how they keep them healthy and working well. And I want to emphasize that I don't have time to give like a full-fledged research talk here, but I want to mention that the mechanisms that we have discovered, including the ones that I show you today in terms of how these energy machines come together, these mechanisms are actually also present in our own neurons. So we're learning fundamental aspects of biology by using these tiny worms that are relevant to how our own brains work. And then we have colleagues, we have friends that can go to other systems, vertebrate systems, even human cells, and test if what we're discovering is actually important to how our own cells work. But more important than what we have learned are the questions that remain unanswered, right? I think as scientists, that's what we do. We think about questions that remain unanswered. And I just want to emphasize that. I want to finish here with my kids again. This is they're like picking up sea slugs in the beach, and the way that scientists learn is by the same way that everyone else learns, which is by asking questions. So the main advice that I have for all the body scientists out there is, this is how we make advancements in science, through curiosity-driven research. And my main advice to the body scientist is don't stop asking questions. It doesn't matter if you're 8 years old or 80 years old. When you stop asking questions, you stop learning. So if the world out there makes sense to you, you're not asking enough questions. Because most of the things out there, we don't understand them. They don't make sense yet. Maybe they're waiting for you to make a discovery. So don't stop asking questions. Stay curious. And with that, I'd like to finish by showing the picture of the people that did the work. This is my lab. They come from all parts of the world. So this is one of the most exciting aspects of being a scientist. I mentioned I'm from Puerto Rico. So Zhao is from China. Sarah is from Pennsylvania. Shavani is from Suriname. Suri, her family is from Korea. Sisi is from China. Agustin is from Spain. Titash is from India. Noel is from rural Pennsylvania. Ian is from Texas. Josh here lives in many parts, but he's originally from rural Georgia. June is from Korea. Richard is from Canada. Lynn is also from China. Leighton is originally born in Jamaica, born and raised here in Connecticut. Mark is from Utah, and Ernesto is also from Puerto Rico. And I've had people that have come from 5 different continents, like over 15 different countries. And one of the most wonderful things about science is that this is actually it's a human instinct. Curiosity is a human instinct that all humans share, and it's a wonderful way of actually connecting with people from other cultures and other parts of the world to find answers to questions that all humans wonder about. With that, I'll finish, and I'd like to take any questions you might have. Thank you.
Questions? Well, not everybody at once.
Yes.
Why does the bacteria have to have sex?
So why does a bacteria have to have cells? So bacteria are actually single cells. They're unicellular organisms. In our case, unlike bacteria, we have like billions of cells. And in our brain, we have 100 billion of them that are called neurons. So there's a difference between the bacteria and us in that the bacteria only has a single cell. Yet the bacteria has inside that single cell, there are a lot of machines that make the bacteria work. We have all of those machines that the bacteria has, plus we have billions of neurons doing it at the same time. Thank you for that question. Any other questions? Yes?
How do we double our lifespan like the past, like, because is it 40,000, within 40,000 years our lifespan had changed, is it like past 10 years just instantly?
Right. So great question. So how is it that for 40,000 years of human history, the lifespan of life expectancy of humans essentially didn't change, and then in the last 100 years it has more than doubled? And the reason is because in the last 120 years in Western medicine, we have had fundamental discoveries like, for example, microbes. So if you don't know about microbes, you are not going to look for antibiotics. And if you don't look for antibiotics, then you're not going to be able to cure infections. So you answered the question, right, that Washington died of a throat infection. And before, if you will have an infection, you will get things like scarlet fever. You know, you can read old books and they talk about these people having scarlet fever. How many people here have had scarlet fever? Nobody, because scarlet fever is not a thing anymore, because you can take antibiotics. Before, if you got an infection, that would be really scary. Actually, most people in war died of infections. So, but that's an important lesson for all of us there. People have wanted to cure diseases for thousands of years. But if we don't understand the root causes, if we don't understand the concepts like microbes, we cannot cure it. We might want to cure Alzheimer's, but if we don't understand how memories form or how neurons function, how are we going to cure it? Our aspiration is not enough to take us to the goal line. And then if you look at the history of science, the way that those fundamental discoveries that we need to make advances in medicine or in technology happen is through curiosity-driven research. Okay. I'm going to share one more story. Like, everyone sees the sun going around the earth, right? Everyone sees that. Every day, you see the sun going around the earth. So for most of humanity, people thought, well, the earth is the center of the world, of the universe. Because you can see, I see it with my eyes, the sun going around the earth. But what did scientists do? Like a scientist called Copernicus, he slowed down and he said, wait, I see it, but is what I'm seeing the truth? Is the sun really going around the earth? Or is it that from my perspective, it looks like the sun is going around the earth and it's really us that are moving around the sun? And he used science to examine what most people were thinking to be truth, to examine and show that it was false, that it's actually us moving around the sun and not the sun moving around the earth. And that's what scientists do. We ask questions when other people stop asking questions, we keep on asking questions. And we examine our assumptions. And that's how we can double life expectancy. When other people are putting masks like this with a bunch of perfumes, scientists are like, wait a second, is it really bad air that's causing malaria? That's the difference. Yes?
So you mentioned that there are certain cells in development that are destined to be a particular tissue, for example, but yet they say some cells are pluripotent can
Right.
come in. is it that those cells are destined to become a particular tissue regardless of their environment or chemical gradient or location in the embryo?
Right,
or
right.
Like how does that work?
Right. No, that's a great question. And one of my best friends is here in the audience, and that's exactly what she studies, Valentina Greco. She studies how is it that cells decide what they're going to become. And it's a very active area of research right now. Like, what is it that tells a cell to become a neuron? That's an unanswered question. In the animal that I use, the programs that tell cells to become a gut cell or a neuron or a skin cell are similar to the programs in our own cells, but one big difference is that they're inflexible. So in the C. elegans, in the nematode, those cells they don't have a lot of flexibility. They don't come out and say, well, what is the environment? What do I become? But in our own cells, the environment actually plays a very important role. That's one of the discoveries that my friend had made and her colleagues, is that the environment plays an important role in telling the cell what it's supposed to become. So the cells have more flexibility. But in all animals, there are programs that tell the cell what to become, and some of those programs tell it to die. Great questions. Any other questions? Remember, don't stop asking questions. Yeah.
So my question is to that. You have some curious scientists in your family, and you seem to be still as curious as when you were that big. How do you keep asking questions? Is it
Yeah.
innate, or
Is it
something
innate?
in your upbringing?
No, I think, how do you keep asking questions? And I think, I think that one of the reasons I kept asking questions was because my parents didn't make me feel awkward for asking questions. So they encouraged me asking questions. And also they felt comfortable saying, I don't know. And I think it's important to recognize that a lot of the questions that you, the kids in the audience, might ask, we might not know the answer. And that is good. That's not a bad thing. It's good to recognize what you don't know. That's actually one of the big jobs that I have as a scientist, is to differentiate what I know from what I don't really know. And so just keep on asking questions until the person says, I don't know. And if they get annoyed by you asking questions, that's their problem, not yours. So you should keep on asking questions. Don't be shy, because when you stop asking questions, you stop learning.
You might think that you have to be really smart to be a scientist. Scientists are much smarter than anyone else. The sociologists studied this, and they found out, much to our dismay, that we actually aren't smarter than the rest of the general population. But you know what we are?
Curious.
Curious. There's another thing, is that we don't know what frustration is. And so you might think an experiment went wrong because it didn't give you the answer you wanted. A scientist goes, that was unexpected. That's interesting. And they keep going at it, right? They never get frustrated, right? And it's that sort of drive to always, find out what the actual answer is that differentiates itself as science.
How are brain cells formed in the beginning before they become destined to be something?
So the question is, how do brain cells form? That's a great question. How do brain cells form in the beginning before they're destined to be brain cells? And the thing to understand about that is that at the moment when every living being in planet Earth started, like each of us, for example, at the moment when our lives started, we were single cells. And that single cell divided a bunch of times to form the cells that will then decide, I'm going to become a nerve cell, or I'm going to become a gut cell, and things like that. All these cells come from this one cell that's capable of becoming many things. And that one cell, then there are a number of divisions where the different daughter cells specialize in becoming specific tissues. But what are the instructions by which that happens? People don't understand yet. And the reason I keep on saying what people don't understand is because when I was a kid and I went to the classroom, they would always tell me what people understood, but they never told me what people didn't understand. So I got the impression that all these questions had answers. And that when I didn't know the answer, it was because I didn't know the answer, and not because somebody else didn't know the answer. Now that I'm a scientist, I realize the opposite. Most questions people don't know the answer, and only the few questions that people know the answers are the ones that are taught in school. So it's important to know that, because if you want to be a scientist, you might get the impression Well, what am I going to discover? Everything is discovered, and that's not true. I see a question about in the back over there, another one over there.
We're going to do the wrap.
Yeah.
If you said what's the middle of the universe, what's the middle of the galaxy?
What is in yeah?
Milky Way.
What is in the middle of the Milky Way? And I yeah. So what is in the middle of the galaxy? We don't know. We don't know what's in the middle of the galaxy. The Milky Way we don't know either. We just like actually in the history of humanity, in the scientific history of humanity, it was only recently, so to speak, that we discovered that that the sun is not going around the Earth, that we are going around the sun. So there are a lot of questions out there that we don't know the answer to, and that is exciting. Lola. How many cells multiply each day? That is a great question for which I don't know the answer. And I think probably some scientists will have estimated it, but I bet you no one has actually gone in and counted every cell that dies and multiplies each day. But I will guess there are millions of cells, because only in our gut and in our skin cells we're constantly shedding cells. Like when you get wounded, for example, new cells form. Like if you get scraped like by falling from the bike, new cells form and replace them, and that just represents the fact that our skin cells are constantly replenishing themselves.
Okay, so I think we'll stop at that point. And I'm sure Daniel will answer questions afterwards, but let's thank Sam for a great talk.
Thank you.